Hello and welcome back to PW English, and I'm your Diksha ma'am. In the previous lecture, we have completed one half of human health and diseases, and today we will be covering the other half of this chapter. So, all those who have not attended the first part, I would like to tell you that we have covered all the diseases thing there. We have covered the viral, bacterial, protozoan, and this AIDS, cancer, everything was covered there. So today we will be discussing about the immunity, vaccination, lymphoid organs, drug and alcohol abuse, the other part of chapter and the most interesting part. So let's get started with the immunity. So what is immunity, guys? So you all have heard of this word in your lower classes, right? So immunity is your defense system of the body. The immunity is your defense system of the body. So you come across a lot of viruses, a lot of bacterias every day. So do you always get a disease, or you always get sick? No, because your body has an inbuilt mechanism wherein it defends your body from these viruses or microorganisms or bacteria or anything. So you have heard, a lot of the thing these times during COVID, that those who have stronger immunity, they are more able to survive towards this virus. So immunity is basically the ability of your body to fight against the pathogens. It's your body's ability to fight against the pathogen. So it is basically the protective system given by nature to your body. So what is immunity? It is the ability of our body to fight against pathogen. So you already know there are WBCs in the body. They are the soldiers of your body, and they are helping us in protecting against the pathogen. But the immune system doesn't finish here. It is a whole lot system, more complex than the other system. It involves a lot of cells, certain proteins, the different mechanism, and the most important, the interesting mechanism, right? So on the basis of these mechanism and understanding all the process of immune system, nowadays vaccinations have been formed and you have been protected from this virus known as COVID, right? So hence we are going to start with this entire thing which is known as immunity. So if I talk about immunity, immunity is of two types. One is a specific immunity, another is a non-specific immunity. So one immunity or immune system is the one uh, which you already have in your body when you were born, right? So you are born with that system in you. Whereas another system is the one you get that immune system after you come across the pathogens after you are born. So one system, when you entered this world, you already had that, and another which you eventually produced in your body, another immune system you produce in your body when you come across all the viruses. Fine. So likewise, immunity is of two types. Immunity is of two types. As I've already told you the words, one is your non-specific, which is also known as innate or congenital. So whenever we are talking about the word innate, congenital, that means they are inborn. They are present in your body when you were born. Whereas another one is your specific immunity, also known as acquired or adaptive. Fine. So you have two types of immunity. One is a non-specific, another is the specific immunity. So let's talk about these immunity one by one. So we'll start with the non-specific immunity, also known as congenital or the innate one. So this is the one which is present since birth. Present since birth. So this immunity have no memory system. Now what is this memory? For example, you have had chicken pox once in your life. A lot of you must have, I think, suffered from that. But you always heard that once if a person gets that chicken pox in a life, that person will not get it again. Why? Because your body produces certain type of memory cells, but this type of immunity do not produce those memory or memory cells, so it has nothing to do with that. So while chicken pox is producing those memory cells, so it is somehow related with what? It is somehow related with the another part of immunity, which is your specific immunity. So since here is no memory, nothing is involved. So this immunity is also specific in, non-specific in nature. How? This immunity will not identify whether the pathogen is a virus, is bacteria, is a protozoan. It doesn't know anything. Whatever comes its way, it will just stop it. So that's why its name is non-specific. So it's non-specific. It cannot recognize the pathogen. It cannot recognize the pathogen. Fine. So if I'm talking about the non-specific immunity, we have two lines of defense in non-specific immunity. One is your first line of defense, and then you have the second line of defense. Now what are these defense or line of defense? For example, this is your enemy. This is your enemy. Okay? And this is you, your body. So your body's non-specific immunity is going to protect you by two means. First of all, if a pathogen has to enter in your body, it has to pass through first line of defense, and then it has to pass through the second line of defense. If it passes through these defense, only then it will enter into your body, and then the other immune response will work. Now, what comes under the first line of defense? In first line of defense, we have two types of barriers. One are physical barriers. One are physical barriers, and another are, guys, physiological barriers. Another are physiological barriers. Fine. In second line of defense, we have some other things. These are the barriers which are cytokine barriers. We will be discussing all of these in detail. Then we have cellular barrier. We have cellular barrier. Then we have the inflammation. So these comes under your second line of defense. So we will be discussing the innate immunity in detail now by discussing these physiological barriers and these barriers of your second line of defense. Fine. Okay, moving further. Let's discuss the first line of defense. In first line of defense, the first barrier we have is a physical barrier. What about the physical barrier? So as we have discussed, there are two types of barrier. One is physiological, another is physical. Whenever we are talking about the physiological word, whenever we are talking about the physiological word, that means something is performing a function. As a result of it, the pathogen has been killed. Okay. Second is physical barrier. That means the presence of something is not allowing the pathogen to come through. For example, there is a wall. Okay, this is a wall. So here is a gift, and here it is you. You want to go there and have your gift, but for that you have to cross this wall. So a wall is present here in between your gift. So it's a kind of a physical barrier that is separating you from your gift. But what if this wall is electrical? So that's a kind of a physiological barrier. That means the wall is performing a function. It has a certain property that will kill you. Okay. Just like that, if this is a pathogen and this is you. So if I have this epithelium as a physical barrier, or epithelium's mucous membrane is there, and this membrane is having a lot of mucus here. So it's a kind of a, it's a kind of what? It's a kind of your physical barrier, or just simply your skin. Your skin is a physical barrier. But what if I say the skin's gland are secreting lactic acid and it is killing the bacteria? Then skin is performing a function, it is secreting lactic acid, it will be your physiological barrier. Fine. So skin is a physical barrier because it's like a wall. But when the skin is secreting something and it is not allowing the bacteria to grow, then it is a kind of a physiological barrier. Similarly, the mucous membrane in the stomach is a physical barrier. Whereas the HCl in the stomach wall has a mucous membrane, and also stomach wall is secreting HCl that is killing the pathogen. It's a physiological barrier. Wherever the chemicals are involved, that's usually the physiological barrier because they're going to perform certain function. Whereas physical one, their presence is just creating a barrier. Okay, so let's talk about the physical barrier. What are the examples we have discussed? The physical barrier, we have skin, and then we have mucus coating. Mucus coating on the epithelium. So that's the epithelium of your gut, respiratory tract, your gut or GIT, gastrointestinal tract, and in fact your urothelium, the one in your urinary system. Okay, so there the mucus will not allow these pathogens to go inside your body, so they're protecting you. Then in the physiological barrier, as I've told you, the HCl in stomach, lysozyme in saliva. The saliva do have lysozyme that kills bacteria. Lactic acid of skin, lactic acid in the skin. These are the type of what? They are the type of your physiological barrier. Simple. So if your pathogen somehow manages to cross these type of barrier, now it has to face the second line of defense, as we have discussed. This is a first line. For example, it has crossed the physical and physiological barrier. Now it's time to face the second line of defense, which contains certain proteins, your cells, etc. So let's talk about the second line of defense. Second line of defense. In the second line of defense, what do we have? The first barrier that we are going to discuss is the cytokine barrier. Cytokine barrier. Listen to me very carefully, that's important. These are not cells. These are certain proteins. What are these? These are certain glycoproteins. So these are proteins which are secreted by virus infected cell. What are they secreted by? Virus infected cell. For an instance, if in your body cells, this is your body cell, okay, these body cells, we are talking about one cytokine. Cytokines are proteins which are secreted by your infected cell. Now these infection can be virus or anything. For an instance, we are taking an example of a cytokine named interferon. Okay, interferon is an example of cytokine, and what it is basically, it's the glycoprotein. Chemically, it's a glycoprotein and it's the example of cytokine. So for example, these are your normal body cell, and some cells somehow gets captured by viruses. Some cells somehow gets captured by viruses. So this is your normal body cell. This is virus. Okay. Now after virus, this normal body cell captured by the virus. So this become the infected cell. Which cell? Infected cell. Whereas the surrounding cells, they are completely normal cells. They are not surrounded by or captured by the viruses. So now these cells, they are like very good friends of their adjoining cells or neighbor cells, so that they want to take care of them. So now what the infected cell, what they're going to do? They're going to secrete. They are going to secrete interferons, which are glycoprotein. Now these virus infected cell, they will secrete this This, this glycoprotein. Now this glycoprotein will bind to the cell, and it will tell the cell that it will, you know, this is a type of a signal, alarm signal. It's an alarming signal by the infected cell to the normal cell. Now these cells, they will get to know, they get alarmed that yes, virus has entered into the body. Now we don't want, or we will not let this virus to enter into us. So it's just like, for example, you are living in a building, and on the ground floor people got to know that one lion or cheetah has entered into your building. Now what that person will do, it will run away to the upper floors and scream loud, the cheetah has come, the lion has come, the leopard has come. Now what you guys will do, you have not come across this cheetah or lion, what you will do, you will close your doors. Just like this, these cells, they are screaming the virus has come. How they are screaming? By secreting the glycoprotein. And now they will bind to normal cell, and normal cell will shut their doors, and they will not allow the virus to enter into them. All right. So these interferons are glycoprotein. What are they secreted by? Secreted by virus infected cell. Virus infected cell to protect normal cell. To protect normal cells. So these are what interferons are. Okay, so one type of the cytokine is interferon. Let's discuss another type of the cytokine. So the second type of cytokine is interleukin. Interleukin, they are also glycoproteins, but they are secreted by macrophage. Secreted by macrophage. What are macrophage? Most of you must be knowing these are phagocytic cells. The phagocytic cells. So we'll be discussing about them in detail. If you don't know, do not bother here. I will make you understand. Don't worry. These are basically cells of your body that are also the soldiers. Okay, major soldiers. So these interleukins, are glycoprotein, they are secreted by these macrophages. Now these proteins, they bind to your body and they enhances your body temperature, or in short form I'll say these proteins are responsible for fever. These are responsible for fever. So responsible for all right. See, for example, this is a macrophage, and macrophage has pathogen in it. It is trying to eat it. Now, when it has a pathogen, it will secrete interleukin. So this is interleukin. This interleukin will bind to your hypothalamus and other body cells. This will bind to your hypothalamus or body cells. As a result, the body temperature will increase. And this will lead to fever. This will lead to fever. Fine. So this is a function of interleukin, and yes, fever is good for your body when you are infected, because when the temperature is raised, the pathogen will not be able to survive in that condition. Right. So that's about the cytokines, the cytokine barrier. We have discussed two cytokines, the interleukins and interferons. Okay. All right. So moving further to the next barrier of second line of defense, and that's your cellular barrier. That's your cellular barrier. Okay. So in cellular barrier, we have various kinds of cell here. We have various kinds of cell. Let's see. One type of a cell is NK cell. Another type is your neutrophils. And then the discussed already, and we'll be discussing in detail, the macrophages. So we have three types of cells here. One are your NK cell, another neutrophils, and then we have macrophages. So WBC, the white blood cell, WBC, the white blood cell, they have various categories. Like you must have heard of the word lymphocyte, neutrophils. If not, we'll be discussing in body fluid. Okay. So there is a category of lymphocyte, that's NK cell. Its full form is natural killer cell. Natural killer cell. And another kind we have is a neutrophil. It's also a type of WBC. So first let's talk about NK cell or the natural killer cell. So NK cell or the natural killer cell are the type of cell in your body which will recognize all those cells, which will recognize all those cells which have abnormal proteins. Your own body cells sometimes have abnormal proteins. Which cells? For example, these your body cells were infected by virus, right? Now, after attacking of the virus, these cells will show abnormal proteins which are not present on the surface of normal cell. So NK cell can recognize them. So your cells gets abnormal protein during two events. One, if it is infected by something. Second, it gets converted into cancer cell. If you know, if I've seen the previous lecture, your own body cells gets converted into cancer cell, right? So two events make your cell abnormal cell by reflecting different type of protein. One, when it is captured by viruses. Second, it has converted into cancer cell. So in both the ways, in both the events, if your cells has become the abnormal cell, natural killer cell will recognize it and kill all those abnormal cells. So for example, now virus has captured it, abnormal proteins have been formed. Now NK cell will recognize this particular cell and it will kill this cell because these cells are not good for your body, right? So what's the function of the NK cell? Yes, it will recognize abnormal proteins on cells, but not pathogen. But not pathogen. It cannot recognize the viruses and bacteria. No, no, no. But your own body cell with abnormal protein. Example, virus infected cell. And the second thing are your cancers. It kills these cells. Okay. Then next we have our neutrophils. Neutrophils are very much important cells of your body. They are the most abundant WBC. These are the most abundant WBCs of our body. Around 60 to 65% of all the WBCs are neutrophils. These are the first one to come during site of infection. For example, you got inflammation, you get infection. These are the one which will first arrive at that point. Okay. For example, the mosquito bites there. These are the first one to go there. Okay. These are basically responsible for pus formation. They are responsible for pus formation. You got pimple on your face, right? So what that pimple is made up of? It is made up of the dead microbes and dead WBC. Which WBC? Your neutrophil. So they work against pyogenic bacteria like your staphylococcus. That's a bacteria that causes pimples. So it kills pyogenic bacteria. What are pyogenic bacteria? That forms pus. And they are the first one to arrive at inflammation. What about your macrophages? Oh my god, these are so important cells. These macrophages, first of all, monocyte, neutrophil, macrophages. These three cells have a very unique property, and that's the property of phagocytosis. That's a property of phagocytosis. What is a phagocytosis now? Very, very important event. Phagocytosis property, it is a property of macrophage, but also of WBCs. Which WBC? Monocyte and neutrophils. They can also perform this process. Now, what is this phagocytosis? Let's see. Phagocytosis is if I say in definition form, engulfing microbes or cells or dead parts of these microbes or cells. Now what happens here? Now what happens here? For example, for example, if this is your broken tissue, why it has been broken? Because this tissue has been invaded. This tissue has been invaded by the microorganism. For example, here is your enemy microorganism, like a bacteria. Okay, so this is a broken tissue, and this is the bacteria. So this macrophage has a property, like whenever they're at some place or at someone's home, there is the aroma of Maggie cooking. You get attracted to it, isn't it? Right? Or you're in your room and you found out that in the kitchen someone is cooking Maggie. So you get chemically attracted to that aroma. Fine. So just like that, the macrophages also get attracted to these microorganisms known as bacteria. So these macrophages, they will get attracted to them. Okay. These macrophages, what they will do? They will form their pseudopodia like this, like amoeba does. Okay. And it will try to eat it. Now it has engulfed it. Okay. This has engulfed it. This macrophage has engulfed it. Now this structure, after it has engulfed it, is known as a phagosome. It is known as a phagosome. And you all know the cells have one organelle known as lysosome that helps in, killing and dissolving stuffs. It has hydrolytic enzyme and it dissolves stuffs, right? So just like that, when this bacteria enters into the body of macrophage, it will become phagosome. Phagosome is just the food anyone engulf. If it is a bacteria, you will again say phagosome. Okay. Now this phagosome, it gets fused with lysosome. This is lysosome. And they both will form a structure that is a phagolysosome. As I've told you, this lysosome have hydrolytic enzyme. Now these hydrolytic enzyme will digest the phagosome, which is basically a bacteria only. This phagolysosome, it will digest the microbe. And hence you say the oxidative burst or oxidative burst or killing has been done. Because the enzymes are hydrolytic, they will also be using hydrogen peroxide there, right? Superoxide and peroxide ions. And that is why we are calling it oxidative burst. Oxidative burst, why? Because we are using here hydrogen peroxide to kill the organism. So this is what happened during phagocytosis, the engulfing of the microorganism or the dead cell or their parts. And this is how the macrophage, because this is a macrophage, do not forget, this is the macrophage. Macrophage engulf the microorganism and then eat it, and you will be free of that pathogen. Fine. So this was about your cellular barrier, where we have discussed the NK cell, neutrophils, and macrophage. Fine. Next is your inflammation. So the next one is your inflammation. So what happened in inflammation? For example, if this is your skin and a mosquito bites you, and whenever the mosquito bites, it you know uh, insert their proboscis, which is the part of the mouth, and and move up to the blood vessel because it wants to have blood. That's why the mosquito is biting you, because it wants to have your blood. Okay. So while it is injecting the proboscis, biting you. So at that time, some of the parts of this mosquito, like its saliva, it enters your body tissues like this. So now at this point, your tissues are getting broken down. Okay, so what one thing is happening here? The tissues are broken. So the mosquito bites, tissues break. These broken tissues contain mast cells, and mast cell also breaks. So when the mast cell breaks, it releases something which is known as histamine. I've done this histamine in the connective tissue as well. I'm not able to recall, I let you know. We have discussed the different cells of connective tissue, and there I told you that mast cells, they secrete histamine, serotonin, heparin. Fine. So mast cells secrete histamine, and this is a vasodilator. Vasodilator. Now what will happen? The same blood vessel, after release of histamine, its lumen gets wider. Why? So that more blood will enter, because that blood will contains a lot of WBCs to fight. So it's a kind of, again, an immune response. But which type of immune response? It's a type of non-specific, because here we don't know who has entered. Who has entered, we don't know. If it would have been virus, maybe the body has reacted in the same. So that's why, because more blood is entering, this surface appears red in color. Surface appears red in color. Now you know. Now you know why when the mosquito bites, it appears red in. Okay, so that's about the inflammation. So the inflammation also helps in your protective mechanism. So whenever the tissue will break, the mast cell will break, they will secrete histamine, which leads to vasodilation, which will lead to increase in blood flow. There will be more WBCs, and due to increase in blood flow, it will appear in. All right. So this was all about your first line of defense and second of the non-specific immunity, which do not generate any memory, which is not specific in nature. The memory doesn't know whether this is a virus or a macrophage or anything. And this is the one which you already had when you were born. You had skin when you were born. You had mucous membrane when you were born. You have all these things when you were born, right? If a newborn gets bitten by the insect or mosquito, definitely its skin will turn, because it already had this type of immune. Okay, so that's about your non-specific. Let's talk about the specific immunity, which is adaptive or acquired. So this is the immunity that you get after birth, or it is formed after birth. It is specific to the pathogen, and it also generates memory. It also generates memory. Very simple example of a chicken pox. You got chicken pox. The immune system is producing certain things. So it will kill the virus of chickenpox only. It will not kill the COVID virus. That means it is specific to a pathogen. Okay. Again, as I've told you, once a person gets chickenpox, it will not get it again. Why? Because during first encounter with the pathogen, some memory cells are formed. So when the pathogen will arrive again, those cells are ready to fight against them. But again, those memory cells are formed against what? Against chickenpox virus, not against any other. That's why we say it's specific in nature, specific to a pathogen, and it has the memory because it is forming the memory cell. If I'm talking about the specific immunity, what are the major cells to discuss? Important cells. Cells of specific immunity. The cells of specific immunity are your lymphocyte, the most important cell. Though there are other cells as well. I'm not denying there are no other cells, but these are important cells. Lymphocytes are of two types. One are T lymphocytes and another are B lymphocytes or B cells. Similarly, we say the T cell or T lymphocyte or B cells or lymphocyte. Okay. Now, we have two types of T cells. We have two types of T cells. One are T helper cell, another are T cytotoxic. T means cytotoxic, another are T helpers. So if their name is helper, that means they are going to help. They're going to help. All these three cells, they can identify the pathogen. They have the ability to directly identify the pathogen. Out of this, T helper cell have a very important function that it doesn't do the things himself or itself, but it will stimulate the other cells to perform certain functions. For example, there is a friend who will never fight but will poke you. He said you this thing. Oh my god, he said you this thing. No, 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 he should not be telling you. You are the guy, you are the strongest one. And you go and fight with the other. Just like that, T helper cell. These T helper cell, they can identify the pathogen, but it will stimulate the other cells to perform their function. If they are not present, probably B cell will not function. Then we have cytotoxic cells. These are the cells, they will identify the free pathogen around there, and they can identify and directly kill them. Okay. So for example, we have a pathogen and we have cytotoxic cell. It identifies it, it say, come here, I will eat you. Will Okay. It can also kill the cancer cell, and it can also kill the foreign cell. For example, someone got kidney transplant. For example, I got someone's kidney. My immune system's TC cell will identify that's not my cell, that's someone else. Okay. So they will identify it, and so that's different tissue. We call it as graft, which have taken from another person. They causes its rejection. They are rejecting them. You are not our bodies. Go away. You are from other one. Right. So we call it as call it as graft rejection. Whereas B cell, these are the cells, they will also identify the pathogen, but they will form antibodies. And antibodies will kill the pathogen. They are not killing a pathogen, they are telling others to kill the pathogen like bees. They are directly killing the pathogen. They're like, come here, I am the dawn, right? And B cell, they will say, I will not touch you. I will form my antibodies and they will kill. Okay. So they can identify pathogen, right? Identifies pathogen and stimulate B cell to form antibody. Form antibody, right? Similarly, cytotoxic cell, they will identify pathogen and directly kills it. Whereas, 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 it can also kill cancer cells and responsible for graft rejection. Okay. Otherwise, your another type of T cells are T memory cells. They're also T memory cells. Whereas B cells are quite different. Let's see how do they work. Very interesting. How does B cell function? So if it is a B cell, if it is a B cell, B cell has a protein, to which the pathogen can bind. What can bind? Pathogen can bind, and it can identify the pathogen. So this is a B cell. B cell, as I've told you, has a nucleus. Obviously, it's a cell, it will have a nucleus. It can easily identify the pathogen. Once it identifies the pathogen, it will ask the pathogen, come, namaste, and come inside me. I will show you heaven. Okay. But that's a trap. That's a trap. Now, what will happen once it enters? The B cell will chop it. Cell will cut it. Now, see, a pieces of pathogen has been formed. Now, B cell, with response to the pathogen's proteins, response to the pathogen's protein, it will get multiplied into two type of a cell. One are memory cell and another are your plasma. Another are your plasma. Plasma cells, plasma cells will multiply and it will form clones. Multiply and it will form clones, and these clones will later on form antibodies. And the surprising fact is that all these cells will form same type of antibodies, and this antibody are specific to the pathogen. Antibody are specific to this pathogen. For example, you came across pathogen. Which pathogen? Chickenpox virus for the first time. Okay, so let's see. Let's do something. For the first time, when you came across the virus, the B cell identified the virus and take it in, stopped it, and with response to it, it formed plasma cell that will form the antibodies specific to the chicken. This, and also it produced the memories, and also it produced the memory cell. But during first exposure with this pathogen, this all thing was happening and it was taking a lot of time, right? In meanwhile, the pathogen being the strongest here now, because we do not have anything, don't know it, so it's now being stronger, it will start giving you a disease. The first time the response is less intensified. The immune response is less intensified. Why? Because pathogen is doing a better job than us. Why? Because in meanwhile we are recognizing it and forming antibody, the pathogen has already done its work. It has already given you the, right? But when the same pathogen comes second time into your body is already ready because it has memory cell and already formed antibody. Now your immune response will be more intensified. It will work in a better way. So here during second time, the more intensified immune response will be shown. Why? Why? Give me answer why. Give me answer why. Because the memory cell are present here. So this first type of response, you call it as primary immune response. Primary immune response. So the first encounter of the pathogen is known as the primary immune response. Fine. So during primary immune response, the pathogen is more powerful and immune response is less because you don't know it. Okay. So primary immune response is also known as amnestic immune response. And this one is secondary immune response, or you also call it as an amnestic. IR means immune response. Okay, so this is how the B cell works and it forms the antibodies. So now we will step forward to some more detailed parts. So the next property or the next point to discuss is antigen. What is the antigen? If you split the word, you will find out it means antibody generating. Antibody generating. So, is the entire pathogen required? Listen to me and answer me. Is the entire pathogen required to form antibody? Yes, I'm giving you 2 seconds. Till then, I drink water. You think. Does we need the entire pathogen to generate the antibody? So, because I have told you they're the pathogen, so you must be thinking, yes, we need the entire pathogen. But even the small part of a pathogen can also generate the antibody and the immune. Yes, how? The part of pathogen which is capable to generate the antibody, you call it as an antigen. For example, you have bacteria, right? In bacteria, you have studied it has LPS layer, the lipopolysaccharide. Fine. So that lipopolysaccharide layer, layer, is the one which generates immune response. Does the bacteria's nucleus generates the immune response? No. So that's not the antigen. So that particular part of any cell, any you can say the pathogen that generate the immune response is known as an antigen. And after generating the immune response, it will react to that immune. It will fight with that immune response. Only then you will call it as an antigen. So what is an antigen? An antigen is basically it's a part of pathogen. It can be a part of pathogen or complete pathogen. that can generate immune response and react to the immune cell. See? Okay. For example, I have this bacteria, and bacteria have a coating known as lipopolysaccharide. So your B cell will not identify the inner membranes of bacteria, but it can identify this blue lipopolysaccharide layer. So this LPS is an antigen. This antigen can be glycoprotein and glycolipid. Okay, so what it is made up of? Glycoprotein and glycolipid. Glycolipid. Okay, so once the B cell identifies it, once the since it is a B cell, once the B cell has identified the antigen, you know that B cell will generate the antibody. Okay, now the antibody will bind to the bacteria. Okay, like this, will bind to the bacteria like. Now this part which is binding to antibody, this part which is binding to the antibody, you will call it as epitope. So I'm telling you one more time because I know you may get confused, but it's quite simple. So the LPS is an antigen which is made up of glycoprotein, glycolipid, and this antigen is identified by the B cell. So B cell cannot identify the entire cell or entire pathogen, but a specific part of pathogen, and that part is known as antigen. Now it will generate antibody with respect to this antigen. Now this antibody will identify all of this and it will bind to it because they are specific to antigen. Now again, the part of antigen that binds to antibody, you call it as epitope. So what is an epitope? What is an epitope? Part of antigen that binds to all right. Now, so we were talking about antigen, which are foreign antigen. That means maybe of bacteria or some other cell that enter into your body. But what about the antigens present on your body? Do you have those? Definitely you have those. Let's see. Let's see. So another topic is of self-antigen. For example, this is a person. This person has a body cell. Person has body cells. So if, for example, this is a cell of a person. So all those proteins present on the surface of its body cells, they are known as self-antigen. What are they known as? Self-antigen. So these are self-antigen with respect to this person. Okay. If a bacterium enters into its body, so these are foreign antigens. What are these? These are foreign antigens. Both are glycoproteins. These self-antigen will enter if they enter into someone else's body. They will be foreign antigen to that person. For example, if this guy give me its cells, for my body, they will be foreign antigen. If I give this person my cell, it will be a foreign antigen. So all the proteins or the antigens present on my body cells, my own body cells, these are self-antigens. Self-antigens are of different types. They are like MHC1, MHC2, and blood cells ABO glycoprotein. What are MHC? MHC means major histocompatibility complex. So you must be wondering, whenever we give kidney to another person, whenever we are a donor, another is a receiver, how will we get to know that this kidney will work into another person? Definitely we will ask that person to give cells and we will check whether they match or not. You must have heard of kidney match, blood group match, right? So what happened there is, by taking blood sample of a donor, we check these proteins on the cells. If these MHC, if I want kidney of another person, if another person's MHC1 matches with my MHC1, that means I can take its kidney. Why? Because my body will recognize it as an, simple as that, right? If I'm taking someone's kidney and that kidney's MHC1 matches with my MHC1, my immune system will think that are my body cells. So my TC cells will not react against the. Very simple. I think that's clear to everyone. Yes. So if I say these are self antigens, so where do you find these? Like blood groups. Like blood groups. If I have A positive blood group, I will be having the A type of antigen. If someone is having B, they will be having the B type of antigen. Similarly, on our own body cells, all completely body cell, we have MHC. What we have? MHC1. All right. Okay. Give me a second. So my all body cell have MHC, whereas my B cells, my macrophages, they have M. All right. So this is how you need to understand the physiology of this antigen, because if you don't know about this, that's why there were a lot of graft rejections have taken place. Fine. So that's about the antigen and self antigens. Let's talk about the another thing, which is your antibody. We are saying again and again, B cell produces antibody. Now, what are these antibodies? Let's talk about them. Basically, antibodies are glycoproteins. These are what? Glycoproteins. And they belongs to gamma globulin family. So, blood protein are of various kinds. Like we have in blood albumin protein, globulin protein. So, globulin are of three type, alpha, beta, and gamma. So, they belongs to gamma category of. These are basically what? They are the glycoprotein. They are secreted by B cell. Secreted at B cell. Secreted by the. Now these antibodies, they are secreted by B cell. If I see, they are made up of like glycoprotein. How the structure looks like? How the structure looks like? They are basically made up of two heavy chain and two light chain. This is how we write it sometimes. H2L2. H means heavy polypeptide chain. Heavy polypeptide chain. And L means light polypeptide chain. Okay. So there are two heavy chains and two light chain. If we talk about the molecular weight, the molecular weight is around 1,50,000 Dalton. So let's draw it quickly. So I have told you there are like two type of chain. One is heavy, two are heavy chain. So these are heavy chain. These are heavy chains. And they are conjugated with carbohydrate. Why carbohydrate? Why we need carbohydrate here? What it is made up of? Glycoprotein. Glyco means, okay. And then we have the two light chain. So these are light chain because they're shorter, so they are of less molecular weight. And they're stabilized by disulfide bonds. They are stabilized by disulfide. Okay, do remember this. So it's a very common question asked in the exam. What are they stabilized? They're stabilized by disulfide. Okay, then again here there will be disulfide bond. Okay, then there are certain regions in this antibody which are constant. You will find that in every antibody. There are certain regions which are constant and you will find them in every antibody. These are constant regions. And there are certain regions which are different in different. Why? Because as I've told you, antibodies are antigen specific. They are formed specific to the antigen. This is a chickenpox virus. The variable region will be different. So I'm saying there are two type of regions in antibody. One is a constant region. This one. This one is a constant. Similarly, this one is a constant. antigen, whereas these are variable regions. These variable regions polypeptide sequence will depend upon what? The type of antigen. For example, if this is formed against the COVID virus, so this region will be different. Again, if I told you this is formed against the chickenpox virus, this region different. Okay, so this region is variable region, variable of light chain, and this is the variable of heavy chain. Okay, this one is a constant region of heavy chain. This is constant of light. Constant region number 1, constant heavy 2, constant heavy. Similarly, okay. So this is a portion that binds to antigen's epitope. What is epitope? The part of antigen that binds to the antibody. So similarly, we call this region because it is similar or complementary to the epitope, because epitope is the part of antigen, right? Epitope is a part of antigen. So this portion is known as paratope. Paratope, it binds to epitope. So if I say the antigen and antibody interaction is basically paratope epitope reaction, is this point correct? Absolutely. Because paratope, because you feel like this is a feet, and in Hindi we say feet as par, right? So paratop comes from here. This is how I say, but it's not like that. Okay, just to, so paratop is a part variable region of your antibody to which your antibody antigen binds. Okay, and what part of antigen binds to antibody? That's epitope. All right. So this is all about the structure of your antibody. So if I split the entire structure into two halves, I will say the entire antibody is divided into two parts. One region is FAB and another region is FC. AB means it is that portion which binds to the antibody. FC is the part which majorly consists of the constant region, and it crystallizes when you react this the enzyme. When you react the antibody with the enzyme, the lower part will form crystals. Okay, it crystallizes faster. So that's why the C word comes from. One, because it contains constant part. Another, it crystallizes. Whereas AB stands for antigen binding. Okay, so you have different classes of antibody. You have different classes of antibody. What are these? Let's see. So the first class of antibody, guys, we have IgG. G means gamma. This is the smallest antibody and it is most abundant. It is the most abundant. This is so small that it can easily pass through placenta. So this is the one which can pass through placenta. Can pass through placenta. Then we have the second type, which is IgA. A means alpha. This is the second most abundant. So we are writing according to their how much they are present in your body. So this A means alpha. This is the one which is a dimer. So this was a monomer. This is one unit. If we combine these two units, it becomes a dimer. Okay. So it's a dimer. That means these two antibodies will be there and both are connected to each other with the help of another chain, which is a J chain. The J chain. So this is the antibody that you will find first of all in the mother's first milk, that is colostrum. So it goes to the fetus, infant. And second, you will find this in your mucous membrane of every epithelium. Every epithelium like your respiratory tract, your GIT and even your urinary tract. So this is the one which is present in colostrum and epithelium of respiratory tract. urinary tract, and your fine. Okay, moving further to the next type, which is IgM. The third one is IgM. M means mu. So IgM, M stands for mu. This is the largest antibody. The largest antibody. The pentamer. It is the pentamer. That means it will be containing that 5 structure. And this is the first one to be formed during new infection. For example, your body come across any microorganism for the very first time. At that time, the body will form IgM. So, first one to form during new infection. So, whenever there is ABO blood grouping, this antibody is formed. For example, you have blood group A and you accidentally took blood group B. At that time, your body will form IgM type of antibody against your B. Fine. Then we have another category of another class of antibody, that is IgD. D stands for delta. This is the one which is present on B cell, on the surface of B cell. On surface of B cell. So while we were discussing B cell, I have drawn one thing that recognizes pathogen on the surface of B cell. That's this antibody. So it will not leave the B cell, it will always be there on B. Then we have IgE. E means epsilon. It is the like less than 1% in number. So this is the antibody that is formed during allergies and parasitic infection. So whenever any parasite comes into your body or you get allergies, this antibodies quantity or number will rise. So if you want to see whether you are allergic to something or not, just check this type of antibody. If its number is higher, you are allergic. So that's about different types of antibody. Now what does your antibody do? What is its function? Let's talk about the function. First of all, antibody helps in agglutination, because whenever the antigen antibody binds, the process that occurs there is agglutination. Second, neutralization. Because if it binds to pathogen, the pathogen will not be able to do anything. What it wants to do? It wants to harm you. So if antibody binds to the pathogen will get neutral. It will not be able to function. Third thing, it can also cause lysis of pathogen. The large antibodies can actually kill the pathogen directly sometimes. And fourth is opsonization. What is opsonization? It is the enhancement of phagocytosis. How? Who does phagocytosis? First of all, who does phagocytosis? Macrophages. If these are macrophages, these are what? These are macrophages. Now, macrophages have a function to do, that it will kill the pathogen, it will engulf it. Now, what antibody will do is, antibody will bind to the pathogen on FAB region. This is the FAB region, this is FC. It will bind to pathogen on FAB region and it will bind to the macrophage on FC region. As a result, it is presenting the pathogen to the macrophage. Just go and kill it. It is telling the macrophage to come and kill the pathogen, to eat the pathogen. So what is this? This is pathogen. So in a way, what it is doing, it is enhancing the phagocytosis by presenting the pathogen to the macrophage. Alright, so these are the various functions of your antibody. Let's move further and talk about the types of immune response or immunity. What type of immunity? The acquired immunity. Which immunity? Acquired immunity. So acquired immunity is of two types. One is active, another is passive. Alright. So your body encountered with the pathogen. and then the immune response is generated. This type of immunity formed is active immunity. Got it? For example, you come across the pathogen, okay? You got disease. Now an immune response is generated. This is active immunity. Sometimes, not always the entire pathogen, but the part of pathogen can also enter into your body. Again, that's an if it is an antigen, you can generate your immune response against it. Again, the immunity is generated. Again, this type of immunity is active. So in an active immunity, somehow you need to come across the antigen. You have to present antigen to your body system, and then that immune system will form the antibody. Okay. Whereas in passive, what if we take the ready made antibodies and sell? Yes. For example, one, there is a person who has suffered from COVID virus. Okay. And then the virus went into person's body. The body recognized it, generate the immune response, formed antibody, memory cell, and the person gets okay. Now, that person's blood have preformed antibodies. Okay. Now, what if I take that person's blood and take the antibodies from that person's blood and give it to the another person? Now, another person is now getting the passive immunity, whereas the first person had formed the active immunity. Okay, so if I'm taking the if preformed, ready-made antibodies, that's passive immunity. But if I am first of all getting across a disease and then forming the antibodies in my own body, that's the active immunity. Also, during vaccination, the immunity formed is your active immunity. So, in active immunity, we come across the pathogen, we encounter the pathogen or its part, and the body forms antibodies or immune cells. So, you are forming the active immunity, one, during a disease, second, during vaccination. How during vaccination? We will come across it when we will study the vaccination. Passive immunity, we take pre-formed or ready-made antibody. Are given to a person. Example, IgA antibody from mother to infant. So an infant consume mother's first milk colostrum, and that contains IgA antibodies, right? Also, anti-venom serum. Venom means the venom of your snake. So whenever the snake bites you, eventually you will get that poison and all. But if you recognize the snake and you go to doctor, the doctor will give you an injection. And that injection contains antibody that are formed against that venom in horse. So in horses, we form these antibodies by exposing that animal to the venom gradually. And that his body will form the antibodies. We extract the antibody from the body of the animal, and then we will give it to the patient who needs. Okay, so anti-venom serum also contains. Okay, all right. So this is about the active and the passive immunity. Next, we'll see which type of this immunity is natural and which is artificial. If I talk about disease one, when you get disease, this type of this a natural active immunity or the artificial? This is a natural type of the active immunity because it's naturally happening to you. Whereas vaccination is artificially, it's man-made. Similarly, the infant is having mother's milk. It's natural. And this one is made by us. So this is artificial. Alright. So that's about the type of immunity, the active and passive immunity. Let's talk about the very important thing, the vaccine. What are vaccines? First of all, vaccines are pathogen or a part of pathogen that generates immune response and memory cells. So, this vaccine, when we got to know about the primary and the secondary immune response, that in primary immune response, the pathogen come across to our body, or we come across the pathogen for the first time, and we form memory cell. And in the secondary immune response, this memory cell works and the pathogen gets killed. Okay, so when we got to know about this, we thought, why always we want to take a disease? For example, if a person come across a chickenpox virus for the first time, the person get disease, and in the subsequent exposure, the person will not get a disease. What if I expose a person with a dead virus, or just the antigen of the virus? My body will again form the memory cell in similar way. So we came across this principle, or this vaccine idea, through the principle of primary and secondary immune response. So what we did, we took a part, not us, but a person who discovered the vaccine, that's Edward Jenner. So Edward Jenner he's the father of vaccination, Edward Jenner. He formed the first vaccine, that's against smallpox. Okay, so there is a disease called cowpox that happens to cows. So he found out that those people who live near to the cowboys and cowgirls, they never get smallpox. Why? Because he found out that the people who live near to the cows, they get exposed to the cowpox virus, which is a light variant of smallpox virus. So when they come across that light variant, the memory cells are formed, and these memory cell will work against all pox virus. So here, the pathogen that we use in the formation of the vaccine, this pathogen can be live or killed or weak pathogen. Okay, so the only purpose is to form the memories. All right, so this is how Edward Jenner formed the first vaccine. Now this is vaccination. So giving vaccine is vaccination. Another thing is passive immunization. Passive immunization. So we give vaccine as a preventive cure. As a preventive cure, so that a person body will form memory cell and the person will not come across a disease. But what if a person get a disease and a person is not recovering? So at that point, we will give that person the already formed antibody against that pathogen. And that you call it as passive immunization. So once there was a very little girl, she was suffering from diphtheria. So this is how the discovery of passive immunization take place. That girl was suffering from diphtheria. So a person, what he does, he took the blood of a person who has recovered finally from diphtheria, and then he takes out this serum and inject it into that diseased girl, and the girl recovered. From this, the passive immunization have its birth on earth, which says that it's the injection or injecting preformed antibodies. So here in vaccination, it's like we are going through the active immunity, and here in passive immunization, we are going through the passive immunity. All right. So this is about the vaccination. Let's see what are the various types of vaccine. That's important. Okay. So first of all, we have we have one type of vaccine, that is a live vaccine. Live vaccine. Okay, first I'll write the categories. First category we have is of a live vaccine. Second, we have of a dead vaccine. Third one, we have of subunit or recombinant. And then we have toxoid vaccine. Okay, let's talk about them. So in the live vaccine, we are using attenuated or weak pathogen. For example, oral polio vaccine, OPV. This is Sabin's vaccine. We also call it as Sabin's vaccine. Oral polio vaccine. So here we are using pathogen that is quite weak, attenuated. That means by mutations we have made it so much weaker that it will not cause you disease. Or rather we are using live pathogen of another variant, but that can cause similar immune response, that can generate the similar immune response. Okay. Then we have dead or heat killed. By giving a lot of heat, we are killing the pathogen, but we are ensuring that the important antigen should not be killed, or it should not destroy. Okay, because antigens are usually glycoproteins, glycolipids. On high temperature, they can denature, right? But while heat killing, we make sure the antigen should survive. And the example is IPV, injectable polio vaccine. It is also known as Salk's vaccine. Okay, in live we are using the entire live pathogen, but it's weaker. But in the dead one, it's healed killed. Also, rabies vaccine is a type of dead vaccine. In subunit, we are using a part of pathogen. In subunit, we are using the part of the pathogen. Okay, and in the recombinant one, what we do is, in recombinant one, we use rDNA technology. rDNA technology to form the vaccine, like hepatitis B vaccine. If you remember, in rDNA technology, we always need a host to secrete something. So here what we did, we took a gene of the hepatitis B virus, then we put it into the yeast, and yeast started producing its antigen. So we produce this hepatitis B vaccine in the yeast. Then we have toxoid. What is a toxoid? When you treat the toxin with certain things and you inactivate it, that becomes the toxoid. I know there are certain diseases like tetanus, their toxin is the one which is, creating a lot of problems. So why not to use that toxin? Because that's the antigen that is generating the immune response. So there we use inactive toxin, which is known as toxoid, for producing the vaccine. So here tetanus toxoid is a very good example. TT. Yes. All right. So these are the various classification of your vaccine and the concept of vaccination. Now you all know how your COVID vaccine was formed, and how you have taken it and how it's so much good for your body. Right? So you should have it if you are in that age bracket. Okay. All right. Moving further to the allergy. Now, what are these allergies? The another name of allergies is hypersensitivity. Okay. So you come across a lot of people. You say, oh, they are so much sensitive. By just small dust, they get so much sneezing and all, right? Or so much coughing. Why does it happen? What's the cause of allergy? We all are different. We all have different genes. Some people can eat cashew and live happily, and some people can die by eating cashew. Yes, that's what allergy is. Allergy is like a drama queen. Sometimes your immune system becomes a drama queen and started behaving in a different way for certain things. For certain things. So that's known as allergy. So we say that allergy is exaggerated immune response to a particular antigen. And that particular antigen is known as allergen. And this allergen can be anything. It can be a pollen, that it can be the denders, the animal skin or hair, right? It can be food products, it can be your drugs, or it can be the, okay. Now what happened during allergies? Let's see. So when you get exposed to a particular allergen for the first time, nothing will happen to you. For example, there is drug named X. Okay. I consume that drug for the very first time, nothing will happen to me. I will never get to know that I'm allergic to it. But if I consume the drug for the second time, oh my god, anything can happen to me. Even death can occur to me. Why does it happen? Again, primary and secondary immune response has a lot of things to role to play here. Because for the first time, for the first exposure, your body doesn't understand what this drug is. Okay, what is it? It's the immune response. Your body is fighting. Okay, your body is fighting. So for the first time, there are no cells to fight for. So you will not generate immune response. So nothing will happen. But for the second time, if you come across the same antigen, already preformed antibodies are there, and now the immune response will react. As a result, you will not be benefited. Rather, you have to face a lot of consequences. Now, what happen is when you have the allergen for the first time, when you have the, okay, let's make it on the another page. When you have the allergen for the first time, okay, so your mast cell gets sensitized by the formation and binding of IgE antibody, like this. So for the first time in your body, when there is exposure of the allergen or antigen, the IgE antibody is formed. Which antibody is formed? IgE. Now, there is second time exposure. This is first time, this is second time. Now what happen is, all of this allergen, all of this allergen will bind to antibodies, like here. Like these antibodies are there, it will bind to it and it will causes breakdown of mast cell. It's just like these are locks and the key binds it and the lock opens. Just like this, when the allergen will bind to all these antibody for this second exposure, during second exposure, the mast cell will destroy. Now here, I'm writing here, mast cell destroy. And when mast cell destroy, there is release of histamine. And you better know, we have also discussed, it causes inflammation. And this inflammation can be so bad. It can be so bad in people. It can lead to sneezing, coughing, and sometimes anaphylactic shock, where a person can die. How? It will causes bronchoconstriction. Your respiratory tract gets constricted and no air will enter and you will eventually die. So this inflammation can lead to sneezing, coughing, and even anaphylactic shock, which can be fatal. Okay. Now, second thing, if I have to treat this person, what I will give to that person? You must have heard of some certain medicine like citrusin and allegra, like these. These reduces allergies. Okay. So what does these really do? If I know the histamine is the one which is causing everything here, what if I block it? I can do it. So for treatment, what I will give people with allergies, I will give antihistamines. And as I've told you, here the person dies because their bronchioles get constricted. If I give something that will dilate the bronchioles, the person can survive, like adrenaline. Adrenaline dilates the bronchioles. And also these steroids, like in chemical control and coordination, we have done a steroid, that is cortisol. That is immunosuppressant. The cortisol is immunosuppressant. That's the hormone. Adrenal cortex, glucocorticoid, if you remember, right? So what is allergy? It's exaggerated, enhanced immune response. What if I give something that reduces the immune response? By using any of these, we can save a person from. So yes, it can be like from minute to fatal, right? So that's about the allergies. So make sure you should be aware what you are allergic of. Some people are allergic of like peanuts, cashews, and a lot of stuff. Okay, let's talk about another thing, that's autoimmunity. So we were talking about a thing. If a foreign antigen enters into your body will generate an immune response. We were talking about it. Fine. But what if? But What if your own body cells are attacked by your own immunity? Yes, sometimes the immunity fails to recognize your own body cell. Your body, your immune system, okay, immune system fails to recognize the body cell. So if fails to recognize your body cells, it will treat them as foreign and it will start killing it. So this is what autoimmunity is. The immune system kills body cells. So there are certain examples you need to understand. You have to learn it because it's quite common. You will get the question out of it. Like example, type 1 diabetes. In type 1 diabetes, the beta cells of pancreas are destroyed. Like in multiple sclerosis, the myelin sheath is destroyed. Myelin sheath, the one we'll have done in the nervous tissue, right? In the in the rheumatoid arthritis, the joints are attacked by your own immune cells, right? Next, we have the lupus. In lupus, the entire body cells are attacked. Then we have vitiligo. In vitiligo, you have seen people with the patches of white skin in their body, right? Other body cells, they have melanin, whereas that patches do not have melanin. That means the melanocyte, the cells of skin that produces melanin, the skin pigment, they are attacked by this. Okay. Also, we have psoriasis. This is also an immune disease or autoimmune disease in which your skin cells are attacked. So here we have type 1 diabetes, we have multiple sclerosis, we have rheumatoid arthritis, lupus, vitiligo, cirrhosis, and then also we have another one regarding the muscles, the myasthenia gravis, in which the antibodies are formed against ACH receptors on muscle. We have done this in locomotion, if I'm not wrong. Okay, so that's about the various autoimmune disorders. Please do remember it. You have to done it nicely. You have to do it nicely because it's rare, they're important. A lot of time the questions has been asked from this part. Okay, so that's all about the autoimmunity. Let's talk about the immune system in the body. So we were discussing about these are cells and this and that. But what type of system forms your immune system? What are the organs involved? Like for digestion, we have digestive system. Like for respiration, you have respiratory system. So what type of system is associated with your immunity? So that is your lymphoid system. And lymphoid system contains lymphoid organs. Lymphoid organs are of two types. One are the primary lymphoid organs and another are the secondary lymphoid organs. Okay, so we have two type of lymphoid organs. One are the primary and another are the secondary. So primary lymphoid organs are the one in which your important cell, the lymphocytes, they are formed and they get mature. Okay, so primary is the lymphoid organ where lymphocyte are formed and gets matured. Whereas in the secondary one, these are actual site of action. These are the organ where these cells will go and they will fight with the pathogen. They will fight with the pathogen. So this is the site where you tell them, you teach them, this is your cell, this is the pathogen cell. If this pathogen comes, you have to kill it. Just like your college. College, you will study the degree of MBBS. They will tell you these are the disease, how to treat it, like that. But where will you work? You will not work in college, you will work in the hospital. So that's the site of your action. All right. So what are the primary lymphoid organs? We have one, bone marrow. In bone marrow, both B cell and T cells are formed. Whereas in bone marrow, B cells are matured. Then where do the T cell mature? In the second primary lymphoid organ, that's thymus. Okay, so T cell got its name from thymus because here they get mature. And B cell get its name from not our bone marrow, but the bone marrow of birds. From the bone marrow of birds, the bursa of fabricus. Bursa of fabricus. This is the bone marrow of birds. Here, the B cells were discovered for the first time and hence they got its name B cell. Okay. What about secondary? What are various secondary organs we have? We have various secondary organs like spleen, like liver, like nodes, lymph nodes. We have tonsils. We have malt, mucus, associated lymphoid tissue in your mucus lining. Because we have a lot of mucus lining in our body. So around 50% is your malt. Around 50% is your malt. Okay. So these are the various, in fact, appendix is also the secondary lymphoid. So these are the various type of lymphoid organ that we have in our body, right? Okay. So this is about the various lymphoid organ. And how does the spleen works? How does it work? What do you think? So spleen works basically, first of all, you must have not heard where the spleen. Have you? No, I don't think so. So if I'm talking about spleen, where is it? Where it is present? If this is your stomach, here you will have liver. Behind the stomach is present this organ. You call it as spleen. So spleen has a lot of functions to perform, though we are not much aware about it, but it has a lot of function to perform. First, it is graveyard of RBC. Second, it acts as filtering organ. How? For example, if this is spleen, this is the artery through which the blood enters, and this is the vein through which the blood leaves. If this blood is infected, imagine this blood is infected. Infected with what? With what? Microorganism. So if it enters the spleen, the spleen will kill all these pathogen, and now the blood which will move out of the spleen will be non-infected. So in a way, it is acting as a filtering organ. What it is acting as a filtering organ. All right. So this was all about your various lymphoid organs in the body. So let's talk about drug and alcohol abuse. So what does abuse word means? It means misuse of something. That means something you're using is harmful to your body and you're taking it in the inappropriate amount. First of all, we'll talk about the drug abuse. Drugs are of two types. One are psychotropic and other are psychedelic. Psychotropic drugs are those drugs which can either increase or decrease your central nervous system activity. CNS means central nervous system, brain and spinal cord. Whereas psychedelic or hallucinogens are those which can alter your perception or reality. It can cause hallucination. What are hallucinations? It means you see something that is not actually present. For example, if you have taken these drugs, you will feel like everything is rotating, or you can see stars right now in the day, or like this board is speaking along with me, just like that. Okay, this drugs can cause you addiction, or sometimes you can get dependent on it. Okay, dependent means addiction, that means you're not able to live without them. Dependence means your body needs it, you cannot work or survive without. So when a person is taking drugs in an initial stage, the person feels happy, fine. So at that time, person have a psychological dependence. The body doesn't need it, but person feels good, so a person is. When a person leaves the drug or quit the drugs at this, the body will be at no harm. But when the person is taking drugs for a longer period of time, the body gets dependent on it. Now the body will not function without these. Here, the body cells get resistant. That means they will not work without the drug. When a person quit this drug, the person will show withdrawal symptoms. Show withdrawal symptoms. Like when a person quit the drug, maybe the person will nauseatic all the time, vomiting. The person will have a lot of tremors. The hands are shaking, right? And a person will not feel good at all. Sometimes the person can also die. This is how bad. So in psychological, there is no withdrawal symptoms seen. Why? Because the body is not dependent here. But in the physiological, the body's physiology is dependent. That means the working of. So let's see what are the various type of psychotropic drugs. The first psychotropic drug is sedative. We're talking about which drugs? Psychotropic. Sedative is a type of drug that decreases your CNS and you feel calm and you eventually go to sleep. So these are given to all those people who are facing depression and they are anxious. So these are basically anti-anxiety drugs. So what are these anti-anxiety drugs? These are barbiturates and benzodiazepine. Barbiturates have barbituric acid. So this barbituric acid is naturally produced in our body when you sleep. So if a person is so much anxious, maybe the barbituric body gets used and you will not be able to sleep properly. So you are artificially giving that person barbituric. So these are one class which gives you calmness because it reduces. Next psychotropic drug we have is opiate. It is again psychotropic, also known as narcotics. These also decreases CNS activity and they also have receptors on your GIT, gastrointestinal. So these are of three types. One is natural. The natural are extracted from a plant known as poppy plant. From poppy plant. Papaver somniferum. Example, morphine and codeine. Morphine and codeine have medicinal properties as well. It's a powerful painkiller. So these are only drugs. You cannot buy it yourself. They're only given to the doctors. Doctors can use it when a person is undergoing a very, harsh surgery because there's a lot of pain during surgery. The patient, doctor can inject it to the patient. Whereas codeine is used in cough syrup. Cough syrup. Okay, because it helps in bronchodilation. During bronchitis, what happens is your bronchioles get constricted. Want to open it up so that you can, and there should not be no coughing. Then we have semi-synthetic. That means you are converting the natural opiate into into the semi-synthetic form. You are chemically treating it. Example, smack or heroin. The smack is white in color, bitter in taste, and it has no smell. It is odorless. So this is how you can identify. Okay. In the synthetic one, they are prepared in laboratory. It has nothing to do with the natural source. They're completely prepared in laboratory. Example, pethidine and methadone. These are given to the patient in the rehabilitation centers who are already addicted to the opiate. These have no proper addiction or dependence, physiological dependence. So that's why we give it, so that you just cannot make a person ask that on physiological dependence that just quit the drug in a day. You need to keep the body in a state, proper state, and also you have to ask that person to quit the drug. So in meanwhile, the function the drug was doing with the cells, these drugs will be doing, but they will not show any dependence. So these are your opiates. Let's see how a poppy plant looks like. So this is how the poppy plant looks like, and this is a skeletal chemical structure of your morphine. So sometime the question may arrive in a picture form. They will give you this picture and they will ask you to identify the drug, or rather they will ask you identify the drug and then see which statement is correct or incorrect. So you need to see the structures. There are only three structures in entire zoology. One of morphine, another of cannabinoid we'll be doing, and third is of a cholesterol in biomolecule. So there are just three, so I think you can easily identify those. Then we have the stimulants. Stimulants are the one that increases CNS activity. So you feel energetic after consuming. First of all, we have caffeine. Caffeine is present in tea. Tea's scientific name is thea. And coffee. Coffea arabica. Then we have tobacco. Tobacco is extracted from Nicotina tabacum plant. So tobacco is the one, it is either snuffed, smoked, or chewed in any of the way. Okay. So this is the one that increases your blood pressure. Yes, this has receptors on CNS as well as the cardiovascular system. So it affects CNS and cardiovascular. So basically, it increases the blood pressure. But it has a lot of side effects. You better know it causes cancer, it causes emphysema, yes, bronchitis. So all these things are related, dangerous. So never ever have this. Then we have cocaine, also known as coca, coke. It is extracted from a plant that is Erythroxylum coca. If you're taking it in a less quantity, if you're taking cocaine in the less quantity, it will be the psychotropic drug. It will be a stimulant. It will give you a lot of energy. But if you are taking it in the more quantity, it can be a hallucinogen. Can be the okay. So you should know what is your limit. Okay. All right. So that's about stimulants. So this there are basically three drugs in psychotropic: opiate, stimulants, and the one we have done, sedative. Let's talk about the one that alters your perception, the hallucinogen. So hallucinogen, they alter the perception or reality. First is LSD, lysergic acid diethylamide. This is really one of a bad drug. Again, I suggest all of you not to have this drug. Then we have psilocybin. Psilocybin is a kind of mushroom. What is it? It's a kind of a mushroom. So not all mushroom are supposed to be consumed. There are certain which are very much harmful. So this LSD is again taken from a fungus. Which fungus? Ergot fungus. Ergot fungus, claviceps. Then we have cannabinoid. Cannabinoids is extracted from a plant that is Cannabis sativa. So a lot of different part of this plants are used, and its receptors are present the various parts of body. Like their receptors are usually on the nervous system, and its receptor are present in the brain and cardiovascular. So this is a type of drug which is abused, mostly abused by sportsmen, the athletes. Let's see how the plant looks like. So basically, these are the leaves of cannabinoid, and the dried leaves are used to make bhang. So you must have heard in India, bhang is made from the dried leaves of cannabis. Charas or hashish is a raisin of the cannabis plant. Ganja is a dried, unfertilized female inflorescence, and marijuana is a dried flowers plus leaf. And this is the skeletal chemical structure of. Make this sure that you recognize, at least recognize. I'm not dealing with any of the chemical thing in it here. I'm just showing you the structure you recognize because you don't have to draw there, right? So recognize the structure and that's your work is done. Next, moving further to the another hallucinogen. Like we have two plants. One is Atropa, another is Datura. So these two plants, one of this is shown here. So there are two structures in NCRT. One is of a poppy, another of Datura. So make sure you memorize the type of flower. Flowers are little elongated, and the poppy one will be hanging here. They usually give the diagrams given in NCRT. Don't worry. If the pictures are in NCRT, they will give that one. Okay, 99.9%. Then, what parts are used? What parts are used? The Datura seeds are used as hallucinogens, and Atropa's aerial parts. Alright. Next, we'll talk about alcohol abuse. Is that alcohol so good for our body? No, not at all. It's not good, and one should not have it. Then why do people have alcohol? Maybe because they feel good. Maybe in your age, people have it for due to peer pressure, like friends are having, so you want to be little cooler. That's why you're having it. But trust me, there are a lot of side effects of alcohol. First of all, alcohol affects your liver. In liver, it causes fatty liver syndrome. What it causes? It causes fatty liver syndrome. The alcohol in liver is converted into aldehyde, and from aldehyde it is converted into fatty acid. And this fatty acid get deposited in the liver, breaking, destroying your hepatocyte. As a result, you call it as fatty liver. Okay, because fatty acid is getting deposited. Why it is going to liver? Because all the toxins, alcohol, everything goes to liver for detox. It also causes liver cirrhosis. For example, for longer period of time, you are consuming alcohol. Your hepatocytes will destroy and liver get destroyed. So this disease is liver cirrhosis. The liver damage occurs. Gastritis. Itis means inflammation. When you consume alcohol, it causes inflammation in stomach. Causes inflammation in stomach. It can also cause a lot of dehydration. So whenever you consume alcohol, it inhibits ADH. It inhibits ADH. What is ADH? Anti-diuretic hormone that prevents diuresis or water loss. If it is inhibited, a lot of water will go out in you. It affects cerebrum and cerebellum. It affects cerebrum. Cerebrum is usually the one that controls your motor speech area, your ability to think. So when you drink a lot of alcohol, you're not able to speak properly. Why? Because it affects cerebrum, the motor speech area is involved there. And also you just speak anything. Why? Because your mental abilities are not properly working. It affects cerebellum, and this coordinates the body. That's why how does people walk after a lot of consuming alcohol? They just sometimes fell off, right? So that's why because it affects the cerebellum. That's why people just sometimes it fell off. Then it also affects fertility, whether it's a man or it's a woman. In both the cases, it affects the fertility of. So see how many side effects are there of these drugs. It doesn't finish here. Let's talk about sportsmen. Let's talk about athletes. What athletes, other drugs the athletes they misuse. So other drugs misused by athletes. Narcotic analgesics, that means painkillers. Diuretics like caffeine, hormones, cannabis, and steroids. Why do they take steroids? Body to be powerful. What happens if a male consumes a lot of steroids? In that case, male can have a lot of acne on the face. It can show aggressiveness, anger, mood swings. Even in male, it can reduces the sperm count and size of testes. Hence, it affects fertility. Also, depression, and in some cases, there is enlargement in the breast. Then, in the female, it can show man like characters. You call it as masculinization, like facial hair, a voice like a man, mood swings, and even female can have irregular periods. So, somehow, this is not advisable for any person to, right? You must have seen a lot of sports person have to undergo dope test before their game. That's because the people consume it. Why do they consume it? Because that enhances the performance. But again, it's not organic. It's right. All right. Moving further to the question. Okay, so the most awaited one. Cancer cells differ from normal cells as they do not show metastasis, divide uncontrollably, show increased telomerase activity, show contact inhibition. So this is a tricky question. Simple but tricky. Just by adding do not, the question has changed entirely. If I say cancer cells do not show metastasis, this will be correct. They show metastasis. They also divide uncontrollably, and there is increase in telomerase activity, but there is no contact. Yes. So they do not show contact inhibition. So this is absolutely correct that they do not show contact inhibition. So answer is, moving further to next, match the following. Quite simple. Column 1 and 2, you have to match it. First is pneumonia. The pneumonia, typical pneumonia takes place with the help of streptococcus, diplococcus, and haemophilus. So A will go with the third. Ringworm is due to fungus, and that is a type of fungus that is Mycophorum. Typhoid due to salmonella, and plague may be new for you. That occurs due to Yersinia pestis. That's the bacteria. Okay, and here the carriers can sometimes be the flea and then the rats. Okay, so that's the answer. Next is, which of the following is true for opiates? So they're asking about opiates, the drugs. Bind to specific opiate receptor present in a central nervous system and GIT, absolutely correct. Is extracted from the latex of poppy plant, this is also correct. It is CNS stimulant, it is CNS depressant. Okay, so there is a modification. Which of the following is not true? So you have to find not true. Okay, so there is some error. It is CNS stimulant or depressant. You know, stimulants are different: coffee, tobacco, coca. But these are CNS depressants. So, which is not true? It is the CNS stimulant. So, answer is. Alright, next question. Which of the following does not reduce the symptom of allergy? Histamine, antihistamine, adrenaline, and steroid. So, antihistamine, steroids, and adrenaline, they all are given during treatment. So, they reduces the allergies. Whereas histamine, it helps in allergies. So, answer is 1. Next. Which of the following is related to antibody structure? H2L2, H2L3, H4L4, H1L1. Simple. The antibody is formed of 2 heavy chain and 2 light chain. So, answer is 1. Simple. If you have done it. If you have read it, you have attended my lecture, you can easily do all the questions. Which of the following is not a part of innate immune response? Neutrophil, mucus coat, interferon, antibody. Neutrophil are the part of second line of defense, cellular barrier. That's a part of innate immunity. Mucus coat, physical barrier. Interferon, cytokine barrier. Whereas antibody, they are part of adaptive immunity or specific immunity. The answer is 4. They were asking which is not the part. Next is, which of the following, okay, I think that's a repetitive problem. All right. So this was all about the chapter Human Health and Diseases. I'll meet you in the next class. We'll be discussing other chapters which are left of 11th class, I guess. Neural, body fluids, and Till then, practice it. Read NCRT. We'll meet in the next. Tata, bye bye.